by repeated viral epidemics. Always prepared and methodical, Doudna presented slides that suggested ways that they may take on the coronavirus. She led by listening. Although she had become a science celebrity, people felt comfortable engaging with her. She had mastered the art of being tightly scheduled while still finding the time to connect with people emotionally. The first team that Doudna assembled was given the job of creating a coronavirus testing lab. One of the leaders she tapped was a postdoc named Jennifer Hamilton, who, a few months earlier, had spent a day teaching me how to use CRISPR to edit human genes. I was pleased, but also a bit unnerved, to see how easy it was. Even I could do it. Another team was given the mission of developing new types of coronavirus tests based on CRISPR. It helped that Doudna liked commercial enterprises. 3 years earlier, she and 2 of her graduate students had started a company to use CRISPR as a tool for detecting viral diseases. In launching an effort to find new tests to detect the coronavirus, Doudna was opening another front in her fierce but fruitful struggle with a cross-country competitor. Feng Zhang, a charming young China-born and Iowa-raised researcher at the Broad Institute of MIT and Harvard, had been her rival in the 2012 race to turn CRISPR into a gene-editing tool. And ever since, they had been locked in an intense competition to make scientific discoveries and form CRISPR-based companies. Now, with the outbreak of the pandemic, they would engage in another race. But this one spurred not by the pursuit of patents, but by a desire to do good. Doudna settled on 10 projects. She suggested leaders for each and told the others to sort themselves into teams. They should pair up with someone who would perform the same functions so that there could be a battlefield promotion system. If any of them were struck by the virus, there would be somebody to step in and continue their work. It was the last time they would meet in person. From then on, the teams would collaborate by Zoom and Slack. "I'd like everybody to get started soon," she said. "Really soon." "Don't worry," one of the participants assured her. "Nobody's got any travel plans." What none of the participants discussed was a longer-range prospect, using CRISPR to engineer inheritable edits in humans that would make our children and all of our descendants less vulnerable to virus infections. These genetic improvements could permanently alter the human race. "That's in the realm of science fiction," Doudna said dismissively when I raised the topic after the meeting. "Yes," I agreed, "it's a bit like Brave New World or Gattaca. But as with any good science fiction, elements have already come true." In November 2018, a young Chinese scientist who had been to some of Doudna's gene-editing conferences used CRISPR to edit embryos and remove a gene that produces a receptor for HIV, the virus that causes AIDS. It led to the birth of twin girls, the world's first designer babies. There was an immediate outburst of awe and then shock. Arms flailed, committees convened. After more than 3 billion years of evolution of life on this planet, one species, us, had developed the talent and temerity to grab control of its own genetic future. There was a sense that we had crossed the threshold into a whole new age, perhaps a brave new world, like when Adam and Eve bit into the apple, or Prometheus snatched fire from the gods. Our newfound ability to make edits to our genes raises some fascinating questions. Should we edit our species to make us less susceptible to deadly viruses? Yeah, what a wonderful boon that would be.